appeal all the participants to join as early as possible. This one-day conference will have three different sessions, all themed on artificial intelligence and its impact on education and research in pharmacy. This conference aims at a few objectives with the objective to give an create an awareness about emerging role of artificial intelligence in pharmaceutical education and research. We have organized this conference to understand the AI-based approach in drug discovery, formulation development, and preclinical research. It will be one of the more objectives of this conference. To bridge the gap between academia and pharmaceutical industry through expert interactions, that will be the next objective for this conference, and to enhance the research competency and digital skills amongst pharmacy students and faculty members. That is one more objective of this conference. To promote the outcome-based education and research innovations in the field of pharmaceutical sciences, we are confident and hopeful that this conference will fulfill all its objectives. The convener for this conference is Dr. K.R. Biyani sir, who is the campus director of Anuradha Group of Pharmacy Institutes. The patron-in-chief, President Honorable Shri Rahul Bhau Bondre sir, he has encouraged us to organize this conference and provided all the necessary facilities required for the conference. We are also very happy to welcome the dignitary speakers who are going to deliver their expertise on various topics related to artificial intelligence during this conference. The first speaker is Dr. Dipendra Aryan, who will be dealing his expertise, who will be delivering his talk on transforming preclinical research in the era of artificial intelligence. His session will begin at 10:30. We will start with the inaugural session at 10 a.m. with keynote address by the convener, Honorable Dr. K.R. Biyani sir. Thereafter, thereafter, we will begin with the technical sessions, with the first session to be delivered by Dr. Dipendra Aryal sir on transforming preclinical research in the era of artificial intelligence. Then, this session will be chaired by Dr. Gopal Bihani sir, who is principal at NK College of Pharmacy, Khamgaon. After first session, we will move towards session 2. Session 2 will be from an expert who is working in the industry based on AI tools in the formulation development. See, the theme is artificial intelligence, and all the three speakers, they come from different facets of pharmacy. The first speaker is involved with, he has dedicated research in preclinical and clinical research since last 20 more plus years. And the second researcher or second speaker during this conference is Mr. Arun Pandian sir, who is a senior research manager at Novugen Pharma, Malaysia. Then this session will continue up to 2 p.m. and 1 p.m. sorry, and then we will take a lunch break for 1 hour, 1 to 2. That will be the lunch break, and then at 2 p.m. we'll start with the third session to be delivered by a renowned academician, Dr. Anwar Sheikh sir, who is a professor and head of Department of Pharmaceutical Chemistry at Alana College of Pharmacy, Pune. So he is very well known for his YouTube channels, who has over 26,000 plus subscribers, and he continuously delivers his talks on for pharmacy students as well as even for NEET students who are appearing. He delivers his talks on chemistry. So with these three wonderful speakers, we are going to organize this conference and we are going to conduct this conference. So I appeal once again, appeal all the participants and delegates to join this meeting immediately. The session is going to start in a short while. All the participants are requested to join immediately. We will begin with the meeting sharp at 10 a.m. Once again, I request all the participants to join as early as possible. So before we join the meeting and before we begin the sessions, there are a few instructions for the participants. Those instructions I would like to read once again, although they are circulated in the WhatsApp group. The first instruction is regarding registration. All delegates must complete the online registration before joining the conference. The Zoom meeting link is shared only with the registered participants through WhatsApp group created by the conference. After you register for the conference, you can join the Zoom meeting and you have to join. It is expected that you join the Zoom meeting 10 minutes before the scheduled time. So it's a good time to join for all of you. Use your registered full name and affiliation of your organizations as Zoom display name. So your name and your organization's name. Professor Unmesh Joshi, Anuradha College of Pharmacy. So I welcome co-convener Dr. R. Pagore sir for this meeting. Welcome, Dr. Pagore sir. Then the next instruction that I would like to read out is once you join the registered meeting, then ensure that you are with a stable internet connection, working camera and microphone. Use a laptop or desktop for better audio visual experience. Mobile phones only if unavoidable. Then keep your Zoom application updated to the latest version. Audio and video etiquettes that we will have to maintain during this international conference. Keep your microphones muted during the session unless instructed otherwise. Turn your camera on during keynote sessions and interactive discussions if possible. Follow the conference schedule strictly. Avoid unnecessary interruptions during the presentations. And the questions may be asked during question and answer session only or through the chat box. You may put your questions in the chat box during the session even. But at the end of every session, we are having a discussion session for a few minutes. So your questions you may ask during that discussion session, question and answer session. Then, as the question and answer sessions and all the sessions will be guided by the moderator. The recording, screenshots, or sharing of the conference contents is strictly prohibited. Kindly maintain professional behavior and decorum throughout the conference. Now, the most important part about attendance and certificates. Attendance will be monitored through Zoom login and participation throughout the conference. The participants for successful delivery and for successful certification, they will have to attend all the three sessions, which will be monitored through their logins. And after completion of the sessions, all the three sessions, The participants have to fill two links. One is the feedback link and another is the certificate link. So after filling the feedback and certification links, the certificates will be delivered within 3 working days after completion of the conference to your registered email ID. So I hope all the instructions are clear. Dr. Pagore sir, you can unmute yourself. Am I audible clearly, sir? Yes, sir. Please kindly unmute Dr. Pagore sir. Is the voice clear, sir?
Yes, yes, sir. Very clear. Yes, sir.
Okay, okay. All the
You are,
instructions are clear enough, sir?
yes, sir. You are audible. Yes, clear.
Okay, 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 okay. We are waiting for convener, sir. As soon as sir joins the meeting, we will start with session one.
Yes, sir.
Yes, yes. So once again, I welcome co-convener Dr. R.R. Pagore sir, convener Dr. K.R. Biani sir, then all the faculty members, respected faculty members, researchers, student delegates, as well as persons from industry. Uh, Please, I once again appeal all of you to log in with your registered name. I am observing some Xiaomi 2411 DRN. Please change your login name in Zoom meeting. Change it to your registered name along with the name of your organization if possible. So registered name should appear during the Zoom meeting. Redmi 9 is one more there. So please change your login names to your registered names. iPhone. iPhone. Pawan sir, please unmute. Yes. Welcome, Dr. K.R. Biani sir. Sir, am I audible? Pawan sir. So, good morning and welcome, Honorable Dr. K.R. Biani sir. Unmute. Welcome, sir. So, I take this opportunity to
Hello.
welcome. Yes, welcome, sir. Welcome, sir. Very good morning, sir. Welcome, Dr. Biani, sir. I hope I'm audible.
Yes, yes. You are audible, sir.
I'm checking with Dr. Biani, sir. Pawan, sir, please check. Good morning, sir. To me. So, dear delegates, it's the high time to begin with our conference. Within a few minutes, we will be beginning with our inaugural session. So, during this inaugural session, we will have a brief about this conference and the organizers as well. And then, with a welcome address by the convener, Honorable Dr. K.R. Biani, sir, who is the campus director of Anuradha Group of Pharmacy Institutes. We will begin with his talk, sir. So once again, I welcome all the dignitaries, all the delegates to this one-day international conference on impact of artificial intelligence on education and research in pharmaceutical sciences. Please, everyone, please check your connections. More and more participants are joining. We will wait for 5 minutes and then so that maximum of the participants they get a chance to attend the inaugural session as well. So we are waiting some more participants to join this link and then we will start the inaugural session of this one-day international conference. Anuradha Group of Pharmacy Institutes is proud to announce this one-day international conference on a very contemporary topic, which will be the future of pharmacy education and research. So artificial intelligence is going to replace so many things. And today, very welcome, sir. Good morning, Dr. K.R. Biyani, sir, and welcome you on this occasion.
Hello.
Yes, sir. Yes, sir. You are audible.
Yes, good morning, everyone.
Good morning, good morning, good morning, sir. So
Yes.
I take this opportunity to welcome Honorable Convener for this conference, Dr. K.R. Biyani, sir, who is the campus director of Anuradha Group of Pharmacy Institutes, Chikhli, District Buldana, located in Maharashtra. So this one-day international virtual conference we have organized, and the theme for this conference is impact of artificial intelligence on education and research in pharmaceutical sciences. The topic is very contemporary and it is going to be the topic or it is going to be the theme for the education and research in pharmaceutical sciences for upcoming years. So this conference is an international conference where the speakers, out of the three speakers, two speakers are from abroad, one from United States and another from Malaysia. So we will have this international conference with a few objectives. So the objectives for this conference are to create awareness about the emerging role of artificial intelligence in the pharmaceutical education and research, to understand AI based approaches in drug discovery, formulation development, and preclinical research, to bridge the gap between academia and pharmaceutical industry through expert interactions, to enhance the research competency and digital skills amongst pharmacy graduates and faculty members, to promote the outcome based education and research innovations in pharmaceutical sciences. So with these objectives, we have organized this conference. At the outset, we welcome Honorable Convener as well as the Patron in Chief, the Chairman of Anuradha Group of Pharmacy Institutes, Honorable Shri Rahul Bhau Bondre sir, who has motivated us and extended all the facilities required for this conference. He made all the facilities available. I also take this opportunity to welcome the convener of this mega event, international event, Dr. K.R. Biyani, sir, who is the campus director at Anuradha Group of Pharmacy Institutes.
Thank you so much,
Uh,
sir. Thank you so much.
yes,
Very
sir.
good
Yes,
morning.
sir. We are overwhelmed, sir, to declare that there is overwhelming response that we have obtained for this conference, not only through Maharashtra, but through the entire pan India. We have got these responses. We have got a few responses from Gujarat, a few responses from Madhya Pradesh, and so many responses from Maharashtra. Over 70 plus organizations are a part of these institutes and the delegates they have registered for them. We are very happy to announce that many delegates from industry Many delegates from academia and many research scholars are also amongst the participants of this mega conference, international conference. We are very happy to announce that the students from undergraduate as well as postgraduate courses, they will also be benefited by this conference. So, with the permission of Honorable Convener, uh, may I request Honorable Convener, sir, to permit to begin the session, inaugural session, sir?
Yes, sir. Yes, sir. Please, please. More than
Yes, sir.
150
Yes, sir. Yes, sir.
participants they have joined already,
Yeah, yeah,
and
already
it is
joined, sir.
now
So,
5 minutes about 10.
yes, sir. Yes, sir. Yes, sir. Yes, sir. So, so once again, before we begin the inaugural session, I would like to read out the instructions for all the participants during the conference. As it is an international conference, we will try to maintain the decorum of the conference. So, the first instruction is regarding registration. All the delegates must complete the online registration before joining the conference. The Zoom meeting link is already shared with the registered participants through WhatsApp group created for the conference, and it will be shared with the registered delegates only. The registered delegates have joined this meeting, and now for registering you have to register with your full name as you have given in the registration. So please check your login names. Ensure that you are with a stable internet connection, working camera, and microphones. For better audio visual experience, please use laptop or desktop. Please use mobiles only if unavoidable. Keep the Zoom application updated to the latest version. Keep your microphones muted during sessions unless instructed otherwise. Turn on your camera during keynote sessions and interactive discussions if possible. Please follow the conference schedule strictly. Avoid unnecessary interruptions during the presentations. Questions may be asked during the question and answer session only. Or through the chat box, you can put the questions as guided by the moderator or the session chair during the sessions. The recording, screenshots, or sharing of the conference contents is strictly prohibited. Please maintain professional behavior and decorum throughout the conference. Attendance will be monitored throughout the Zoom login participation. The e-certificates for participation will be issued within 3 working days of the conference only to the delegates who attend the sessions and submit the feedback link as well as certificate link both at the end of sessions. I also take this opportunity to welcome Dr. Dipendra Aryal, who is the expert speaker for the first session of our conference. Uh, at the outset, I would like to invite Dr. K.R. Biani sir, the convener for this conference, to give a welcome address and tell a brief about our conference, please, sir. Dr.
Thank
K.R.
you. Thank
Biani
you
sir,
so much. Thank
convener
you.
and
Thank you
campus
so much,
director,
Vishal
sir.
sir. Thank
Thank
you,
you, sir.
respected chief guest, esteemed resource persons, honorable chairpersons, distinguished academicians, industry experts, faculty members, dear research scholars, and my beloved students, ladies and gentlemen, good morning to one and all present over here.
Good morning, sir.
It
Good
gives
morning.
me immense pleasure and a deep sense of pride to welcome you all on behalf of Anuradha Group of Pharmacy Institutes, Chikli, to this international level academic program on artificial intelligence in pharmaceutical education, research, and drug discovery. Today, it is truly a special academic occasion as we come together to deliberate upon one of the most transformative forces of the 21st century, artificial intelligence, and its rapidly expanding role in pharmaceutical sciences. Warm welcome to all dignitaries. At the outset, I extend my heartfelt welcome to our esteemed resource person who have graciously accepted our invitation and spared their valuable time to enrich us with their knowledge and experience. First author, Dr. Dipendra Aryal, our esteemed proud alumni, first batch student, now president at Genial Pharma Care USA. As Dr. Amit Pant could not join today, so I extend my thanks that Dr. Dipendra Aryal, our proud alumni, has consented and he is with us. The second resource person, Honorable Shri Maniam Arun Pandiyam Rajendran, a senior manager, formulation and R&D, Novigen Pharma from Malaysia. And the third esteemed resource person, Honorable Dr. Anwar Sheikh Sir, is a professor and head, Department of Pharmaceutical Chemistry, Alana College of Pharmacy, Pune. We are truly honored by your presence. Your academic excellence, industrial exposure, and research contribution will undoubtedly inspire our students and faculty. I also extend warm welcome to all session chairpersons, Dr. R.R. Pagorit sir, Dr. R.H. Kale sir, Dr. Gopal Bihani sir, very senior academicians, principals, faculty colleagues from various institutions, research scholars, and industry professionals who have joined us today, both physically and virtually. Friends, about Anuradha Group of Pharmacy Institute, as you, majority of you are well known, the Anuradha Group of Pharmacy Institute, Chikli, stand as a premier education group in a Vidarbha region, founded by late Karmayogi Tatya Sahib Bondre and now headed by ex-MLA Shri Rahul Bhau Bondre, with a proud legacy of more than 3 decades in pharmaceutical education. Our group comprises 4 pharmacy institutions affiliated all to Sant Gadge Baba Amravati University, Amravati, offering diploma, degree, postgraduate, and PhD program in pharmacy. Since the establishment of Anuradha College of Pharmacy, since 1994, our journey has been guided by a single vision to impart quality pharmaceutical education rooted in ethics, research, and social responsibility. Over the years, our institution have consistently demonstrated excellent university result, national level achievement in GPAT, NIPER, other competitive exam, and strong on-campus placement, and the biggest that the strength, a robust alumni network contributing to academia, industry, healthcare, and research globally. Dear participant, we are living in the era where artificial intelligence is no longer optional, it is inevitable. AI has already begun to reshape every dimension of pharmaceutical sciences, including drug discovery, molecular modeling, preclinical, clinical research, formulation design, optimization, regulatory science, research data analysis, scientific publishing, etc. What once took what once took What once took years of trial and error can now be achieved in a month or even weeks with the support of AI-driven tools. This program has been thoughtfully designed by Professor Joshi sir to create awareness about the emerging role of AI in pharmacy, bridge the gap between academic and industry, empower faculty and students with digital and research competencies, and to promote outcome-based education and innovation in pharmaceutical sciences. To my dear students, I wish to say the future pharmacist will not only be a drug expert, but also a data literate, technology aware professional. Your ability to understand AI tools, interpret data, and apply technology ethically will define your success in coming decade. To my faculty colleague and research scholar, AI is not a threat to education. It is a powerful enabler. It allows us enhance teaching effectiveness, improve research quality, increase publication impact, and foster interdisciplinary collaboration. Let us embrace artificial intelligence as a partner in progress, not as a replacement for the human intellect. Overview of today's academic journey program is structured into three highly relevant technical sessions, each addressing a crucial domain. Session 1 will explore how AI is transforming drug discovery and preclinical research. Session 2 will provide industry oriented insights into AI based formulation development, and Session 3 will focus on AI tools for pharmaceutical education, research, and data analysis. I strongly encourage all participants to actively engage, ask questions, and reflect on how these insights can be implemented in your own academic and professional journey. Anuradha College of Pharmacy Institute, we strongly believe that education must evolve with time, technology, and social needs. We are continuously striving to integrate emerging technology into curriculum delivery, promote research driven learning, and prepare our students to meet global professional challenges. Programs like today's conference are a reflection of our commitment to academic excellence, innovation, and lifelong learning. Friends, now I take this opportunity to acknowledge the organizing committee. I will like to take place on record my sincere appreciation to the organizing committee, to the all coordinators, co-convener, faculty members and supporting staff, technical and IT team, and especially to respected Professor Joshi sir. Now he has played a very vital and key role to organize this international conference. His efforts made this possible that such a type of the latest that such a type of subject that the need now this particular conference we can now organize. His dedicated efforts have made this academic event possible. A special word of thanks to our management and leadership, whose constant encouragement and vision continue to guide us in organizing such a meaningful academic initiative. Before I conclude, I would like to emphasize one thought. Artificial intelligence can process data, but it is human wisdom that gives it direction. Let us ensure that as we adopt AI, we also uphold the core values of ethics, empathy, responsibility, and scientific integrity, the true foundation of the pharmacy profession. With these words, I once again extend warm welcome to all participants. I wish you highly enriching, insightful, and productive academic experience. I am confident that today's deliberations will spark new ideas, inspire collaborations, and open new pathways for research and innovation. Thank you for your kind presence, patience listening, and active participation. Wishing the conference a very great success. Thank you so much.
Thanks a lot, respected sir. So it were the words from our mentor, our guide, and the convener of this conference, Honorable Dr. K.R. Biani sir, who is the campus director of Anuradha Group of Pharmacy Institutes, such a big group of institutes, four pharmacy institutes headed by Dr. K.R. Biani sir, and marching on the right path under his able guidance. Once again, thank you very much, sir, for your constant motivation, continual support, and creative ideas and effective leadership who has taken up to this path. Sir has very correctly said during his inaugural talk that artificial intelligence is a partner in our progress and AI can AI can facilitate the work, but the human wisdom is the key which gives directives to artificial intelligence tools. And therefore, we are committed, we should be committed to stick up to the ethics, to the morals, and to the empathy and With passionate research, we are going to march with the facilitation by artificial intelligence tools. Thank you very much, Dr. K.R. Biani, sir. With the permission of Honorable Chair, I declare that this conference, on behalf of Honorable Convener, sir, I declare that this conference is inaugurated.
Yes, sir. Yes.
Please declare, sir. Please declare, sir.
Yes. Now the session is open. And
Yeah.
I again take this opportunity to welcome our first speaker, Honorable Ariel, sir.
Yeah, yes, sir, yes. Thank you, sir. Now we are going to begin with the technical session, the most important and a very happening session that with which we are opening this conference is the session one, that is transforming artificial intelligence and drug discovery and preclinical research in the era of artificial intelligence. The speaker, I would like to introduce the speaker before we take over to him. The speaker, Dr. Dipendra Aryal sir, is MPharm and PhD in pharmacology. His educational journey is as as clear and as diverse as we can see during his education. He has completed the undergraduation from India, from our college, and we are very proud to mention that he is the pioneer alumni of his institute, completing his B.Pharm from Anuradha College of Pharmacy. Post his undergraduate education, he has completed his post-graduation M.Pharm from Kathmandu University in 2001 and 2002. Post that, he has completed his PhD in Dankook University, South Korea. See how diverse is his path of education as well. He has completed his B.Pharm from India, post graduation from Nepal, and the doctoral work he has performed at doctoral work he has done at South Korea. During his postgraduate PhD work, he was involved with bioequivalence trials, clinical trials, establishing many important generic medicines. His work during this research was published in Clinical Trial Symposium, and it was reported to Korean Food and Drugs Administration. His educational journey does not stop here. He continued with his postdoctoral research at Duke University Health Systems. He worked there during his postdoctoral work mainly on behavioral pharmacology, and his contribution to Parkinson's disease was Parkinson's disease and many behavioral research, behavioral studies, psychopharmacological studies. The research was published in Nature. We all are quite aware that publishing a work in Nature, it carries the whole introduction of our resource person, Honorable Dr. Dipendra Aryal, sir. Not only with his academics, but his vast work experience of over 20 plus years, he has passionately worked in the field of clinical and preclinical trials. His experience, right now, he is the president at Genial Pharma Care LLC, Baltimore, Maryland States, United States of America. He has also contributed as clinical faculty at Kathmandu University from 2002 to 2003. Then he has done his postdoctoral, he was working as postdoctoral researcher from 2007 to 2014 at the prestigious Duke University, United States. Then he has also worked with UNC Healthcare as pharmacy intern from 2017 to 2019. Then as a clinical pharmacy fellow from 2020 to 2000 till date, I think, at Houston Methodist Hospital, Houston, United States. So the biodata is so impressive that we are quite excited to hear from Dr. Dipendra Aryal. Before we begin his session, I would like to introduce the chairperson for this conference, Honorable Dr. Gopal Bihani sir. He will chair this session. So Dr. Gopal Bihani sir has graduated from NDMVP, the prestigious institute in India, NDMUP Samaj College of Pharmacy at Nasik. Post that, he has completed his post graduation from Bharati Vidyapit Pune and continued for his doctoral work from Bharati Vidyapit Pune. After completion of his postdoctoral degree, he had joined academics, and since last so many years he is associated with Anuradha Group of Pharmacy Institute, and he is shouldering the position of principal NK College of Pharmacy with entire competence. He is also a pharmacologist. Our organizing committee also contains two pharmacologists as the members, and the speaker is also a renowned clinical pharmacologist. So, without taking much of your time, I welcome Honorable Dr. Deependra Aryal sir to open his talk. Deependra Aryal sir, please. Hearty welcome once again.
Thank you, Josie, sir. I'm really excited and overwhelmed because it's been a while, almost like 26 years since I graduated from the ACPC. And I'm seeing quite a bit familiar faces today, so it's more exciting and thrilling opportunity for me. I know I was being called upon on the last moment, but since I have been working in this area for some time, I said this is my quite a good, impressive opportunity to share my experience to my esteemed professors and new students and other faculties. So I'm really glad to see the old and familiar faces again after so many years. Let's get started. So I've been given with the topics to cover uh, preclinical role in drug development and how AI can input. I think my phone is ringing. Just a second, sir. Hello? Yeah, it's good. So I will go through this with a brief introduction of the preclinical stuffs, how we do and develop a drug, and then go through the preclinical. And overall aim is to register to the FDA and then go beyond that. And then I will catch up on how AI, advanced technology, can help us with to navigate through that process. So let's go brief. The drug development process takes long, many years. So it starts with a concept and trying to develop a drug for a specific target, like receptors or signaling molecule or chemical synthesis, and so many different physiological aspects. And once we figure out a target, then we try to screen different chemicals and find a best suitable one which act specifically to navigate or mitigate the disease status. So overall preclinical goal is to find a best molecule and then try it against in vivo or in vitro testing, and then write an application for IND. Because if we work on develop a molecule, our ultimate goal is uh, to market it, to make it, more clinical oriented, and then we can, do good work in the society or make some money. So the main aim is to put the application to IND, and once IND is approved, then we will go from there to develop clinical trials, and then once clinical trial is approved, then we will file an NDA application, and then once NDA is approved, then we go for marketing and we do post-marketing surveillance. So this is the general strategy from how small molecules to IND application, NDA application, then clinical trials, and then post-marketing surveillance works. So I will try to focus more on the preclinical side. So in preclinical side, basically we do first target, whether we are targeting a receptor uh, or pathophysiology, then we have to identify, okay, what is the foundation behind it? And then once we understand the problem and then try to mitigate the remedy on that, then we will try to find a compound which will hit the target, and then we validate it, then optimize it, then we identify a lead compound, and then once we identify the lead compound, we try to optimize it, and once we get the optimization for a desired effect, then we move towards the preclinical development phase. So let's go brief on these each aspect and see where we can use AI or artificial intelligence side by side. So first, target identification and validation. It's also called pre-screening foundation. So the goal here is to identify a biological target. It could be a protein, it could be a gene, or it could be any pathway linked to a disease. So once we identify key activities like disease pathway analysis, target disease association, or genetic and functional validation, then we can use different tools we have currently. Like bioinformatic database, you can use Uniport or GeneCard, to identify pathophysiological sequencing or signaling pathway, whether that will be the good target for our desired research. Then omics platform like genomics, proteomics, those things are helpful. And recent advancement in CRISPR or Cas9 genetic editing, we can splice a specific gene and see whether that gene will have any contribution to the target of our like biological or physiological, pathophysiological pathways. And overall, these things is kind of vast. It's a very intensive research work. And most of the time it is literature review, uh, analyzing the current available data, and those part of things will probably, very labor intensive. That's why, network biology modeling is important. So knowledge of graphs, how signaling is working. So there, AI can contribute. We will go in details later with examples, but you just understand once in the target identification phase, when we need a lot of information available in the current form, which can be raw data or very well established pathophysiological pathway, then AI modeling can help us to process those data quickly, and we can have some idea. Okay, we can identify a target, which disease model we want to touch. This is our target and validation. And then once we identify our target, we try to figure out, okay, what kind of molecule it could be. So that is the goal is find a compound. So because we use a lot of compound to see which will be most effective. I'm not going to touch it here, specificity and sensitivity. That part comes very late, but we just try to hit it, whether it is working as slightly or like 5%, 10%, or 20%, whatever percent it is. Sometimes even 1% can be a starting point. So we try to screen like millions of compounds and then try to figure out that one. So here, there are so many tools. And in current position, this is a very highly developed field because we have a molecular docking system, we have a like HPLC system, we have a mass and LCMS system. So those things are very genuine tools and very important. It can do hundreds of assays at one time. So screening is very important, and then we can get those data and then use it to the virtual screening. So where AI can be important, especially when we do modeling, AI is really helpful because just making one graph and try to understand, okay, what this graph is telling us, it takes a lot of time for a scientist, to really put a thought on it. So modeling, especially getting help from the AI is really important. Then sometimes even we break down the molecule, our target hit molecules, and see whether these fragments will give any idea for a ligand binding or something, So for that, NMR spectroscopy, X-ray, cholecystography, and surface plasmon resonance, there are so many tools we have. So this is basically a generalized hit identification. Once we got the hit, then we have to validate and prioritize whether this is have any good impact or not. It's just a hit or it will have any physiological outcomes as well. So we here confirm activity and remove false positives. So activity we do here is secondary selective profiling, early toxicity checks. And we have cell-based functional assays, counter screen, early ADME assays, and AI-based heat triggering models. So specifically, if we say AI role here, the AI will be very helpful for modeling. So having a huge data set and then using those data set to a specific model will help us to heat validation and prioritization. So once we got a hit compound, which is already validated, then we think about whether this compound will have any drug-like properties or not. I mean, there could be so many things, but if it doesn't look like a drug, like it has to be taken, it has to go through the metabolic pathway, it has a certain percentage of crossing absorption, or if we are developing a psychiatric drug, then it has to cross through the blood-brain barrier. So we have to have some sort of understanding whether these molecules has some sort of drug-like properties. So we look for potency, selectivity, solubility, metabolic stability. Those things are very important on that. So for that, we have to understand the medicinal chemistry of the compound and a structure-based, structure activity relation, and then computational chemistry like QSAR models. And so SAR and QSAR are very easily handled by AI models. So trying to understand SAR even is kind of hard. So for us, even a perfectly trained people will take hours to do modeling, but AI could be helpful here. So once we identify a lead, then we have to optimize it and see whether it can have some sort of safety, efficacy, or pharmacokinetic profile to suit for a drug molecule. So here, ADME has to be evaluated, and toxicity also has been seen. And then the tool we use, ADME assays, silico ADMET predictions for toxicity, and other AI models, toxicity, or X-ray, crypto EM, structure guided design. Those things will be helpful tools. So once we got lead optimization, then we move that compound into the preclinical development. And we know most of the people, because we are in a pharmacy profession, we know preclinicals basically gives us idea about the in vitro and in vivo studies. So in vitro studies, the mechanism of action, cytotoxicity, drug-drug interaction potential, those things, and mainly cell lines, uh, imaging, those things we use for in vitro studies. And in vivo studies, mostly we do PK/PD study and toxicity using a rodent model. Especially these days, because we have so many knockout animal models, like we slice a particular gene, and develop a new mice with that knockout gene. And those kind of mouse model is very helpful to to evaluate a specific target molecule. So using those tools, we will do in vivo analysis very carefully. And if we got the data from those type of studies, AI can help us to synthesize the modeling and figure out whether the drug is according to our perception or not. So what if we just summarize these all effects and process and steps, then we can think about, okay, what AI can contribute. So because the first phase, preclinical phase, is very data intensive phase. We have to work so many compound to get a one compound which is possible to be a drug. And we have to do so many studies to check all possible effect, side effects, unnecessary things. So in those things, AI could be very helpful. Let me try to introduce some of the concept which we use current days. Even we may not use the AI to do that, but we use it for data generation, like bioinformatics. We know most of the disease model because we know genomics of a human is very well studied, and we know a lot of things about the which gene could be important or not for any disease model. So bioinformatics is really huge field these days. So we can hypothesize, okay, based on these bioinformatics data, our goal of designing a drug for a particular disease will be helpful. So understanding bioinformatics is very important. And recently, organ on a chip is also developed. It's still ongoing, but small single organ and single PK modeling has already been on market. But organ on a chip is very important because it will reduce the use of animal directly, and it's a chip also. So using that organ on a chip, we can generate a pharmacokinetic data very quickly, and we can feed that data into the AI system, to get a better understanding of PK. And there are high throughput screening, which is very useful to finding a hit and finding ideal compound. And then this is a PDSP. It's a specific. It says a psychoactive drug screening system. So here, what happens is there are so many receptors in the body, and there are so many psychoactive compounds we know. Then we try to screen our targeted or design compound against whatever available in current form. We run through the system, and then we can identify, okay, this target compound is affecting so many which receptors it's prioritizing, which is it not prioritizing. So these tools are very important to generate the data. And once we get the data, then we can use this data to go further and decide, okay, we are on the right track of developing a good molecule or not. So I will summarize the current scenario of AI drug discovery into three different forms. So one is structure-based drug discovery, where AI will be very helpful to understand ligand binding and where the compound is fitting into the structure. Other is uh, analyzing the target molecule against a huge chemical database which is currently available. The AI will be really helpful for docking, understanding a receptor docking and a chemical database, using a chemical database, and also the ligand-based drug discovery. So I work in developing one novel compound, which I think it's best suitable in this forum to discuss how we work on. So basically, we do is we try to figure out because analgesic like morphine. Morphine has very ideal properties for analgesia, but it also have a side effect, like it could cause constipation if you take for a long time, and it may cause a respiratory depression, and people may die. So, like we know these days, a fentanyl issue is very high. People die taking the fentanyl. So we try to work on understanding mu receptor of opioid and try to figure out whether we can create a new compound which only target good things, like it will produce analgesia, but it will not produce side effect like constipation or respiratory depression. Let me go brief to separate good and bad, highlight the good and bad effect of the morphine, how it causes. So usually when morphine bind with the mu receptor, mu is opioid receptor, it go through two signaling pathway. There are so many pathways it go through, but I will try to focus on only two. So one is GPCR, which is a good thing because it produces G protein coupled receptor signaling pathway, and this is a pathway which is basically causes analgesic effect. And there is a second pathway, it's called beta-arrestin pathway. The beta-arrestin pathway, when activated by morphine, it causes constipation and respiratory depression. So if we design a molecule which target more biased towards the GPCR, but do not activate beta-arrestin pathway, it means we will have a compound which will produce analgesia, but will not cause any side effect. This is very interesting concept. So we work on, here is also Nobel scientists, Nobel laureate are here, like Brian Kovalka, who got the Nobel Prize in working on the GPCR, and he got it on 2012 or something. And he developed a receptor, and then we work on the zinc library, which has like 50,000 compounds on it. So we use zinc library to target mu receptor and see which compound will have a preference towards the GPCR, but will not activate beta-arrestin pathway. So it's like 50,000 out of 50,000 compound, we got one hit, just one hit. So we call it compound number 7. But compound number 7 has some issue with the solubility, and perfectly locating into the receptor pocket. So we have to modify that compound and get a best suitable compound which will fit into the GPCR pocket, and then it will have a bias signaling towards the GPCR, but not activate beta arrestin. So this is kind of things where AI can contribute. We published this paper in 2016, and I think in 2016 there is no hype about AI, and people were just doing the computational thing. They didn't have idea whether we can have a AI to do the more complex data analysis and have their own brain to get even the outcomes by the computer. So we work, this is the extensive project we work. So we had a 3 group. One is focused on designing molecules. Other was focused on checking in vitro data, creating in vitro data and see whether this compound has a bias signaling or not. And third group was to check whether this compound have any in vivo effect or not, animal testing or not. So I was involved in the third part, like preclinical studies in the animal model, and I did extensive work on checking whether it has an analgesic effect or not, whether it has a side effect like constipation or not, whether it has a side effect like respiratory depression or not. So overall, we able to find this compound very suitable and has some effect on analgesia, but very little effect on constipation and respiratory depression. And we were able to publish it in Nature in 2016. That was my dream. When I was in ACPC, we used to talk about what gonna do when in the starting days, I used to say, "Oh, I want to be biochemistry scientist," but I remember Dhawale sir was laughing at me. So you don't have to be don't have to study pharmacology, just pharmacy, to be a scientist in biochemistry. So because of that incident, I was more interested towards the receptor and pharmacology. And so my ultimate dream was to publish paper in Nature or Science. So I was really happy on that time when we were able to publish in the Nature. So we already went through a little understanding of how drug development works and how pharmacological uh, knowledge and expertise is important for generating the data. Once we get the data, then we can use AI tools to process those data more effectively and get us sincere outcomes as possible, as quickly as possible. So if we summarize the role of AI, how it can contribute, it will say it will accelerate the discovery of novel therapeutic agents because it will shorten the screening time and finding a good hit, you know. So that's very helpful. And also it will improve accuracy of prediction because AI can easily model. Uh, so if we provide AI with a lot of physiological data or receptor data or ligand-based data, then can model and give us perfect idea, okay, this molecule is best suitable for further research. And because the AI computing power reduces the overall time and cost associated with the drug development. So we will discuss further these things with a more robust example. For example, we compare like fluoxetine Prozac, which is developed by Eli Lilly, to compare with the Zyprexa, olanzapine, developed by Eli Lilly. And recently, AI designed molecule is TNIK kinase, is a novel compound for aging and fibrosis, which can be used for aging and fibrosis. So let's compare these three compounds. So the target of Prozac is serotonin reuptake transporter, and olanzapine is dopamine and serotonin GPCR, and reton, rentosertib. It's a mouthful of words. So this is for TNI kinase. So the one works on transporter, other works on GPCR, and the other one works on kinase. And this discovery style is Prozac is more like a traditional phenotypic and functional assays, and we check a compound against, like I described earlier, against the functional enzyme, functional things. And olanzapine, we do SAR activity extensively because of we know the receptor, target-based receptor. And so for return search is like AI-enabled target ID and molecule, and based on the molecule structure, it will figure out, okay, this is more closely related with the binding pocket of the receptor or not. And if we compare the timeline, Eli Lilly took almost like 15 to 16 years to develop this drug, whereas based on having a more robust receptor data of serotonin and dopaminergic receptors, Eli Lilly was able to develop Zyprexa in 5 to 7 years. So now, because of so many data is available in a data set, uh, this new molecule AI can be easily, AI has a very easy way to model it and against the available data set, and it developed in like 2.5 years. And it's already in the phase 3A trial. So it already got IND approval, NDA approval, and it's already in the more robust clinical phase. And if we compare the cost, is a very different. Like first, project costs like millions of dollars. Of course, olanzapine also costs millions of dollars, but because of the time, low time, it reduces the cost significantly. And for rentosertib, it is very low cost because we usually do computational everything in the computer. So there is less manpower and less time. So it is very uh, less costly compared to others. This is kind of gives us a timeline for this molecule, how quickly it evaluated from using a biology AI to understand, okay, what is the uh, which receptor we have to target for getting a novel compound which target aging and fibrosis. So they choose, based on the available knowledge, they choose TNIK pathway and use the model. They started in like 2019, early 2019, and they use the data set against the chemistry AI, medication AI, and they kind of find a, a lead compound, and in within a year or so. And then they use in vitro assays and get the pharmacological profiling of this compound. And then soon after, they use animal studies and were able to file the IND, and then they go in clinical trials. It's a very, very quick process. If we compare the timeline, so they started target discovery, AI target discovery in 2019, and then they generate molecule and optimize it within a year. They went to preclinical candidate nomination like after validation and everything in February 2021, and then started studying in 2021. In 9 to 12 months, they were able to submit IND. So they already in post IND phase, like doing the human trials in 2021. So we can see how quick AI can produce very robust discovery, identifying compound to move to the clinical phase very quickly. This is the first example of AI discovered target demonstrate that AI can be compressed target discovery, medicinal chemistry analysis, preclinical timelines, and even set a new benchmark for AI-enabled drug development for future. So by going through that example, we can understand how important AI becoming for the future, because by day goes, we have more understanding of receptors, or even post like post signaling pathway of a drug, and that data will getting used day by day. And if we can use AI to target a molecule against the available data, then we will be really quick to develop a very suitable molecules. So usually target identification is first step where AI can significantly contribute. So we have a input, which is available data like genomics, proteomics, metabolomics, so many data set is available. And AI can compute these data and then use the medicinal chemistry information and to find out a best suitable compound which can, work on that particular receptor or particular enzyme. And then use that available data set information and see, uh, uh, compare it with the available paper or published articles or clinical trials resources, and see, okay, we are going to the right track or not. So this is if we are not using a human brain to judge, okay, we are going right track or not, because in advancement of machine learning, AI can even decide, okay, I am going in the right track. So even a layman turning on the computer and processing all the data can get a better understanding. Okay, AI is telling, using their computational power and machine learning, okay, this is molecule is going to the right track. It is working very well against a particular receptor, particular enzymes, particular pathways, and it has supported by available resources, clinical data set, clinical trials. So we are in right track. And once it gives idea of, okay, what's going on with the right track, then we can use our human brain to, to test on the disease and the drug ability and toxicity and novelty, and we can figure out ultimate target identification. So I know AI seems to be very uh, encouraging for future, but there are so many things still we have to figure out. So hopefully in future there will be a better understanding. We just started developing a new drug uh, using the AI, but there are other area where we need to more polish to get better data set, develop a better data set to get a AI input. So like for example, drug design and lead optimization, internal new molecular structure can be searched against a huge library, but we have to build our library as well. So usually library is built, putting like hundreds of compounds in a like testing tubes, and then we analyze it and see whether it has a signaling or not. So virtual screening is a very entirely important for AI, because we can't do virtual screening. AI can do, because it can do deep learning to simulate billions of interaction, identifying hits in a week rather than years. So it can simulate very well using the deep learning. And AD met toxicity based on the available database. It can figure out which drug is have a very good ADME profile, and whether it has a good efficacy, therapeutic outcomes, or it may have a toxicity effect. Hope I'm in the right time. So this is a kind of basic understanding of how we can use a mole prophet. This is also one AI tool which can handle virtual screening, AI molecule generation, AI-based molecule, AI docking, and even analyze like drug interaction, drug likeliness, synthetic pathway, and a structural alert. So this is tool under development. So the Rotencity, I gave the example of, they usually use mole prophet AI tool to get development that drug. So if you are interested on further study, there are so many paper published. For example, this is published in the medicinal chemistry. It gives how AI assisted drug discovery pipeline can be rapidly yield drug candidate with clinical potential. AI-driven target identification to assist AI molecular design. Employed a generative AI-driven de novo design to rapidly design novel analogs. So basically, AI is emerging. We just started to use it for complete work out the whole process, work out for designing a drug. Hopefully, in future, we may use more AI to shorten the time and the cost of drug development. I think that's pretty much I can tell on how AI can be used in the preclinical setting. If you have any questions, please let me know.
Uh, you can also elaborate on clinical development also,
Clinical
which is also.
development, right now, AI is not that much helpful for clinical development because clinical development is very regulated by FDA. So there is nothing AI can predict. But AI is using in clinical data set. Once we do the clinical trial, then we get a huge data set, a patient profile, and what happens is usually we get a like we go for blood testing in any hospital visit.
Right.
We do all unnecessary testing. Like we do sodium, potassium level testing, everything. We like A1C testing, every lipid profile. So those the data set is usually not handled by human. They just set aside for further review for future if any question arise later in post-marketing surveillance. So those data set were kept aside, and only main data set is focused to justify whether drug is working or not. But AI has been used and those data set to uh, to simulate whether any output is coming. So AI is mainly in clinical studies used for computational power, but not machine learning. So in future, probably machine learning is also given to AI, but I don't know. FDA has not authorized yet. So maybe in
Okay.
future more machine learning will be used in the clinical trials and probably, I mean, the thing is, what could happen if we have a fully AI developed system? So the best thing is we need very low number, very few people to test a hypothesis. So like for clinical trials, we use like hundreds of patients. But if we have a AI tool, then we may do it in like couple dozens of people. So time and resources will be very less, and we will get a very good data as well. Usually, the number of people, how many number of people we need to test a hypothesis is determined by the deviation, how much deviation we're getting
Statistics.
on the data. But we narrow down the deviation using the AI resources, then probably we will need low number of people. So that's a very effective area where AI can contribute.
Okay. Okay. Over to Dr. Biani, sir. Dr. Gopal, sir. Session chair.
Yes, sir. Uh, First of all, I would like to really express my sincere thanks to Dr. Dipendra Ariel, sir, for delivering
Thank
such
you,
an
sir.
impactful and insightful session based on the very recent trend of using the artificial intelligence in the most backbone of the drug discovery, that is a preclinical research. Really, sir, the session was very informative and it was really highly enriching. Uh, as we all know, sir, preclinical research basically forms the backbone of any drug discovery. And
Yeah.
compared to traditional ways of preclinical research, which were too lengthy and costly, but definitely, as sir has insisted, that AI use is going to help us to reduce that time period, and also it will be helping the scientists to ensure that the new therapies comes to the market which are more safe and effective compared to the traditional methods. Sir, I would just like you, can you please let me know ki What reliable are the AI-based toxicity and efficacy prediction compared to the traditional methods which were used previously for by using the animal studies?
Yeah, thank you very much. This is really important question because toxicity prediction is kind of hard because we root out possible toxicity when we do the hit compound and try it to model against the receptor and other things. We do like PDSP analysis, everything. We always try to lower, like rule out the compound which may have any other interactions. So for toxicity analysis, there are different kind of tools is coming which can do in vivo analysis in a plate. We don't need animal tissues and other things. And there are also there are some tools which is using chemicals to possibly check possible toxic effect. So AI usually can help us to model the physiological aspects and to check whether any deviation is going on and possible toxicity can happen or not. So in summary, AI will help us to predict toxic effect. But in general sense, in the old days, we we were only able to predict like physiological based on physiology and pharmacology. We could predict the toxic effect, but any deviation from the standard physiology, pharmacology, we would not able to predict the toxic effect. So usually toxic effect, adverse effect, we would have seen in a post-marketing world. But now, because of the AI and the very robust modeling, we understand much easily the toxic and adverse effect.
Yes, sir. Yes, sir. Rightly, sir. Sir, looking at the current scenario of drug discovery, sir, so many drugs were developed from the past so many decades, which we have even the history. Many drugs got approval from regulatory authorities. They were launched in the market, starting from thalidomide disasters and even recently the most tragedy which took in United States itself, the rofecoxib tragedy, the drug which was launched by the Merck. I think it was banned later on, and it caused the deaths of thousands of patients in USA also. Sir, how AI can be useful in such cases where the drug is even approved by regulatory authorities and it has got approval in the market. So how AI can be useful to predict the toxic effects in that cases, sir?
So, because AI has a machine learning ability and it can model robustly and quickly, uh, even things which we usually ignore, AI can focus on those things as well. So, based on that, most of the serious side effects are supposed to be well known ahead of time because it is either pathological based or chemical based or even receptor based. So because of the robust modeling and machine learning, AI can be used for understanding any possible serious side effect which may happen in the future.
Okay. So one more, sir, what skills today's pharma professionals, especially let it be faculty members, students, or even the industry pharma professionals, sir, what skills they should adapt uh, to look forward to these, to remain relevant in this AI-driven pharmaceutical world in the coming scenario? What skills these pharma professionals should learn, sir?
So in any scientist, basically, even if we compare from the old generation. So using the available tools and interpreting the data, those are two key things to develop in scientific world. So the students should have a robust understanding on how the process is working through drug development and how your knowledge can contribute in each aspects of the drug development. And because basic understanding is always important, so learning and robust and having a robust understanding and in like ADME things may provide a better understanding in the future. So like when I was in a pharmacy student, we always had a very wonderful session when we start learning on like therapeutics and even pharmaceutics. And my best interesting subject was biophysics when I was a student. So if you, Joshi sir probably laugh at me. So, having a basic understanding, we used to teach each other understanding of receptor is very important. So having a basic understanding will uh, help a student to navigate through all these processes. So I would suggest them focus on the basic concept. I mean, of course, the exam is always important. Getting a good marks is important. But if you understand the basic concept, then you will be successful. So AI is just a computational power and machine learning. Nobody will, even AI will not abolish our ethics and our understanding. So just, finding a job will be difficult. But if you know the basics and you can use the data better, then yeah, you will be successful.
Yes, sir. Yes. Thank you, sir. Thank you, sir. Thank you so much, sir. I would like to invite questions from participants, sir. Sir, if any of the participants would like to interact with honorable speaker.
My question is there,
Mhm.
sir, there is any possibilities in the future that AI completely replace the human scientists in the drug discovery?
I mean, there is possibility, but it depends on us whether we develop enough data on every receptors, every pathways, every disease models. If we only keep generating data and feeding to the AI, then yeah, AI will be more powerful because AI will work on the data generated by us. So I will not say AI will replace us, but it's a plot of movie, whether AI will kill us all one day or not. But I don't think AI will replace us, but AI will make our life very difficult. In the sense, if you are a new student and trying to get your foot in the pharmaceutical world, then you will have a hard time finding a job because most of the jobs will be done by AI. But if you are well known in your field, then I don't think AI will replace us anymore. But yeah, I mean, it could be pessimistic to say AI is really important, but just finding a good drug molecule is not the ultimate answer. In a drug development process, there are so many steps. Passing the ball through all the lines. I don't think AI will ever do their own. Our input will be always important. So I don't want anybody to be pessimistic on that. We should have be always optimistic. Our mind is superior than AI.
Thank you so much, sir. Thank you.
You're welcome.
Any more questions? Any more questions from audience, delegates? Depends, sir. Can you put a light on your C7 discovery a bit more? Discovery of C7. Yeah, yeah, yeah. How that timeline, that I want to just get more emphasized
C7.
from you. C7, your
So,
compound.
oh, this one.
Yeah, 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 yeah.
So, Dr. Kovilka got the Nobel Prize. Let me see when he got it. Oh, he got a Nobel Prize in 2012
Okay,
in chemistry with Dr. Robert Lefkowitz, which is also faculty in the Duke University. I didn't work with him, but he was next to my lab. So both scientists I know, and I work with Dr. Kovilka. So they developed GPCR in
okay. That,
early 20, like 2004 or 5. And since then, they extensively working on beta arrestin pathway. So Dr. Lefkowitz kind of hypothesized in 2004 or 5,
I
beta
got it.
arrestin pathway is the main pathway which is important to create a side effect of morphine. So that was
Okay,
known. And then Dr. Dr. Kobilka were able to crystallize mu receptor. So when they got the mu receptor crystallized, now we know which pocket it binds, morphine binds. And from there, which pathway will go. It's a GI coupled pathway or beta arrestin pathway or any other pathway. So then we got a better understanding of mu receptor around
okay,
2010.
okay, okay,
So after that, it took almost 2, 3 years to get a better understanding of a possible compound with a very high selectivity in the GPCR pathway and low selectivity in the beta arrestin pathway. We call it like it's kind of more like a biased agonist.
okay,
So it took almost 2, 3 years. And once we got that compound and it shows some in vitro better in vitro data testing, and then we finally decided, okay, let's purify this compound and get the better structure. So we had a basic understanding of PGM7 compound, which is working like we decided or we
okay,
want. It took almost like 2 years to come up to the PGM21.
okay,
So basically, if we ignore the crystallization of receptor and finding a beta arrestin pathway, after that, everything developed. It took like 2, 3 years to get the compound concept, and
okay,
then
okay,
it took. I was fully engaged in this project for 2 years, and I was
okay,
only engaged in like almost like when we got the PGM21, and then I was engaged. So it took me 2 years to validate in vivo and in vitro data. So it
okay.
overall this project cost us like 4 or 5 years.
Fine, fine. Hello. If it would have been the era of AI, it would have taken much shorter period of time.
Yeah, yeah. Like
Right,
first part, the finding a better hit, yeah, I would have done like in like within like 3, 4 months.
right, right, right.
And after if now medicinal chemistry data is so robust now,
Mhm.
even we will have a better PGM21 structure compound, and any chemistry we will can develop that compound using that information. So
Okay,
I
okay,
think the first step will be very short.
okay,
But
okay,
the
okay,
modeling, yeah, and animal testing will still take time. So I think overall, I will say within 1.5 years, we would have be able to figure it out.
okay, okay, okay. Thank you. There are
Mhm.
few questions, few more questions in the chat box. I request Dr. Pawan to take up those questions from
Can AI
student.
improve the clinical trial monitoring and data analysis?
Yeah, data analysis for sure. Uh, Clinical trial monitoring, I'm not sure uh, what you are suggesting there. So clinical trial monitoring is like mm, because we don't know in clinical trials whether people are taking drug or placebo.
Mm.
So it is mostly double
It's
blinded.
blinded, blinded.
Yeah,
Yeah.
double blinded even.
Right, right, right.
So even scientists, a doctor doesn't know whether he's taking placebo or a drug.
Mhm.
So it is kind of hard to do clinical monitoring at the time. But yeah, data for sure it will do.
Okay, thank
Um,
you.
One more question is there. Is there any technique for the students to learn molecular talking from like AI?
uh, there is couple of software, but mostly they are uh, in like proprietary software. This
Okay.
is very expensive. Mm. So I don't think like a general student, will have access to that software right now. But down the road, probably yes. But there are public library will be coming soon. I mean, for GPCR, there are public library in NIH.
Okay.
NIH has public library for GPCR. So there you can understand a docking mechanism as well. But for general receptor chemical docking. But medicinal chemistry docking, I don't know if there is free library or not.
अजून घे म्हटलं. दोन्ही घेऊन टाक क्वेश्चन्स आहेत अजून. One more question I think is there. What type of AI used for drug discovery? Probably I think whether generative AI or any other type of AI.
Yeah, I mean, right now we say generative AI, which is working with a have with having a machine learning capacity. That is widely used nowadays, but it may be some other uh, AI, could have been developed later. It usually, I will say, in drug discovery, there are a few distinguished things, like I showed in one slide. One is ligand based, other is GPCR based, other is chemical based. So I
Right.
believe in future also there will be different domain of AI which will be focused on particular work, and that could be used in a particular concept.
One more question is focusing on how AI can use patient selection and personalized medicines. Personalized medicines seems to be the focus.
Okay, how AI can use for personalized medicine?
Personalized medicines.
Yeah, like we ask usually Chat GPT for personalize our resume.
Right, right.
Yeah, basically, personalized medication is, as we know, is a main concept that medicine should be focused on individual people,
Right,
because
right.
if we go deeper, we'll say, okay, if individual people has a genetic profile, so some people
That,
may
that,
have
that.
some drug effect better, some may not have. Usually for some drugs it is, but most of the drug genetic profile is not that important. Uh, yeah, I mean, if we kind of start developing a AI for individual patients, give a domain, specific to any patient, and give data of all the medication or disease condition given to that patient, then probably AI will be helpful.
I, I
I mean,
think
even our
probably
phone has some AI, thing, and he can remind us, okay, Mr. Dipendra, today is your meeting, okay? So that
hmm.
kind of information AI can easily provide us.
Okay, 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 okay. I think these are the most of the questions are covered, and it was really a very very mind blowing session. And at the outset, I would like to thank you for sparing your family time on this weekend, as well as keeping awake late nights for us. Really, it was a very pleasant session with you, meeting once again as a expert speaker. You have put a very clear light on the entire scenario of drug discovery and what can be the possible role. probably AI has to come up yet in full fledged. So maybe we have to imagine rather today. Uh,
Yeah.
am I right? So we have to imagine what may be the possible role of AI. But your session was very, very enlightening, and we are very proud to see your research. Just one small request from many participants is there. If you would be able to share this presentation with them. So
Yeah,
if you share
sure.
it with us, we will be dropping it in the form of a PDF in their WhatsApp group.
Okay, sir, sure, no problem.
Yeah, yeah, yeah,
And I would like to thank you, Dr. Amit, as well, because he provided me slides, Uh,
yeah, yeah, yeah.
even though I came late in the picture, but his slides are very helpful for
Right,
me
right, right,
to
right.
to
He was
come
unable
up.
due to some unforeseen circumstances were there, and so he was unable to take up his session. Maybe it is reserved for him some other time. Maybe.
Okay, that's good.
But yeah, 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 yeah. So you have begun the conference in a really international manner, putting a light on entire things in the preclinical as well as clinical, and your own experiences you have shared. As an alumni, this would boost the morals of our ACP students also, as well as all the student delegates who are over here. Many researchers are also there in the amongst the delegates. I hope there is a possibility of coming out with some collaborative research. Some of the delegates may request you for some collaborative research in future also. So I thank you. Really, I am overwhelmed to see you as a speaker and the depth of your knowledge and expertise. Thank you, really,
Thank you, sir.
Dr. Dipendra.
If I can say a few words to the student of ACPC,
Yeah, yeah. Please, please.
So I would like to address to the students of SCPC. I was the first batch there, and studying at SCPC on those circumstances were very different. Now you are probably more privileged where the robust campus environment and the structures are developed, and you have much more resources there right now. But when I was just there, our all resources were our teachers. So, just think about it. I studied there in SCPC on those days, and now I was able to work with a Nobel laureate in the USA. So, so you are in good hands. Just focus on a study. You are really in good hands, and they are very robust teacher, very intelligent. Even I appreciate. Even I would teach anywhere. I follow their way of teaching, so I learn from them. So you are in good hands. So be focused and be and have a fun time studying and dedicate your study time. And I appreciate everyone for attending today, and really I'm thankful to Joshi sir for giving me this opportunity. So you are muted.
So express my very sincere thanks to the session chair, Dr. Gopal Bihani sir, for chairing this session, moderating this session in a very pretty good manner. So it was a pharmacology dedicated session. The pharmacologist was an expert speaker, as well as the pharmacologist was the moderator of session, the session chair. So it was very fantastic for we Sutix people to enjoy that session. Thank you. Thank you. May I now request Dr. Pavan sir to release the certificate, appreciation certificate for Dr. Dipendra, please. So, Dipendra, we are looking forward for your visit, physical visit to your campus.
I wish, sir. I'm also, hoping to meet
Yeah,
everybody. So
yeah, yeah. Please, please. Ready, sir? Just hold a minute, please. How was your experience?
it, for this seminar,
Yeah, yeah, yeah.
yeah, it's really a wonderful experience.
So,
I mean, I had some anxiety because I was presenting in front of you.
so
So
it was really great to hear from you. Thank you. Pawan sir, ready? Yeah. So once again, we appreciate Dr. Dipita Rajal sir
Thank you, sir.
for everything that he has done today, and looking forward for many more of such conferences and your talks and any more thing that we can do for our students as well as the college. Thank you very much,
Sure.
Dipita sir.
Thank you.
Thank
Thank
you.
you
Thank
very
you
much,
so
sir.
much,
So nice.
Dr. Ariel sir, for such a informative sessions. Thank you, sir.
Thank you. Okay,
Thanks.
bye.
Thank you. So that was session one for our one day international conference, and it was really a nice opening session. Uh, I would request Dr. Pavan sir to check whether the okay. So Dr. Manian sir, the second speaker is also with us. Dr. Arun Pandian sir, he is the second speaker. The session two of our conference, one day international conference, is going to begin shortly. The theme for the conference is artificial intelligence, impact of artificial intelligence on education and research in pharmaceutical sciences. So, my dear friends, the first session it was directed towards one area of pharmaceutical research, and that was preclinical and clinical research. Now, once the clinical research is over, simultaneously the researchers go for product development. So that is the big area where there is a great contribution from the researchers to develop a product. And therefore, the second session is designed in a specific manner. And the title for the second session is Artificial Intelligence for R&D and formulation development especially. And an equally important and equally proficient speaker is available or he has consented for this session. Before we begin this session, I would like to introduce today's expert speaker. Today's expert speaker for session 2 of this conference is Mr. Arun Pandian sir. Mr. Arun Pandian sir is a senior manager formulation development at the renowned Novugen Pharma Malaysia. It is a renowned pharmaceutical company in the Asian region, as all of us are aware. Arun sir has over 16 years of extensive experience in pharmaceutical research and development. He currently leads a team of formulation scientists, engineers, and technical staff who is responsible for end-to-end product development from feasibility to the end step of commercialization for global market including USA, European Union, Malaysia, and rest of the world. Prior to joining Novugen, he was associated with three reputed pharmaceutical organizations, Stride Sassoon, Ipca Laboratories, and Glenmark Pharmaceuticals, where he played key roles in developing and launching complex doses form. So, first session that was contributing towards the research for drug development, new drug molecule, and now the second session is contributing towards the research in formulation development. He has successfully developed and commercialized over 35 products. Really, it's mind blowing, including modified release tablets, pellets, bilayer tablets, suspensions, and hot melt extruded micro suspensions. His expertise covers QBD and DOE based formulation design, bioequivalence study support, cost optimization, and first time right technology transfer for 20 plus global launches. Arun has a strong track record in regulatory dossier preparations like ANDA, ASEAN, as well as European Union. So, patent design around strategies and lead the teams in formulation development. He is also an award-winning contributor for many aspects in formulation development. So, such an expert speaker is available for us. to deliver the session 2, that is AI in formulation development and formulation research. Dr. R.H. Kale sir, MPharm PhD, who is the principal of PRMSS Anuradha College of Pharmacy, who is a PhD in herbal drug technology and herbal formulation development is his expertise area. So, an equally good chairman to chair this session. He will deliberate the session and he'll be the session chair for session 2. So, without taking much of your time, I invite Honorable Arun sir as well as Honorable Dr. R.H. Kale sir to take up the session. Dr. Arun sir, please, you may start with your session, please, sir. And thank you very much. Before conclusion, also, I would like to thank you as well as the management of Novogen, especially Dr. Mr. Irfan sir, for providing such a great resource person for this session. Please, sir.
Thank you so much, Umesh sir, for a wonderful introduction. I was overwhelmed with the introduction, and there was an quite intense session, first session, which I was patiently hearing on the preclinical side, uh, which in our research matters a lot, even though we deal with a generic, but the basic science remains the same for any molecule or any development. That was a quite intense and interactive session. I really appreciate and I like the most. So I'll keep on the energy as well as the momentum, whatever I have prepared for my dear colleagues as well as the researcher who are attending and listening to me. So just confirm me where once my presentation is visible.
Uh, Presentation is not yet visible.
Okay.
Please start sharing slides. You are visible and audible quite clearly, but
Okay.
presentation is not yet. Yeah, now it's visible. Now it's visible.
So I'll summarize from the last presentation of the preclinical session whatsoever on the drug during the preclinical session, how it's tracked to the different receptors and how machine learning as well as the large language model will intercept that together to make a quick turnaround for any molecule to know how does it going to impact in the body or on each human being. So once that study is done, and then finally the formulations or finished dosage forms, which is available in the market. Now, what my role over there to support Novogen as a formulation scientist is to quick turn around my generic formulation so that I can benefit the society or the people who is really in need of. Now, how we are using this technology, AI technology, specifically when I meant here the drug discovery is not an intense way what a previous speaker has said about a preclinical session, but drug discovery over here, what I meant here is a different kind of a molecule, salt or different processes that we meant for an formulation development. So as a formulation scientist, always in a generic market, we will have one challenge that if any of the innovator challenges the generic not to use certain things with the patent. When I say certain things, it can relate to the drug of its polymorphism form or particle size or a process, packaging, anything lead to the certain patented, and it's linked to the formulation development. Once that prerequisite is challenged, it means it's going to overall link to the formulation development, which can drag or delay the development in a generic field. And then it can lead to the in having a lot of resources, utilization of money, and then finally it's ending up, nowhere in the business, which ultimately leads to a business, but finally we cannot serve to the society. So in this presentation, what you can expect? You can expect what is an AI currently in a formulation development is taking place and how it's evolving this in a formulation development. and what are the advantages that we are grabbing from this AI. What is the key thing which is different from the traditional way of development, and what is evolving so that we can quick turnaround using an AI. And I will go through some of the current available workflows of AI on a live session, so that researchers or the students can understand, I don't know, how it has been built up right now and how it's going to evolve in the future as well. And I will be touching about the different AI model, how it has been utilized, why I am currently talking about an LLM or NPL, and then how it's going to be utilized in a longer perspective so that it will be integrated into a deep learning system. Then what is the utilization that we are doing so that the formulation development can quickly turn around. And as a student or as a researcher or as a pharmacist overall in the industry, how should we get ready to utilize this wonderful tool and ourselves to prepare ready to modify our way of thinking so that we can rightly utilize our skill learning right now, and so that we can adopt to these changes. And I really enjoyed the question answer session, and in case if I have anything, then I'll be taking up. So the first thing which I said about what is an AI and advantage of an AI, probably using a large LLM model like ChatGPT or Gemini, you can understand that how the AI working for you. But the thing is here, for a pharmaceutical industry, I will say that earlier it was a chunk of data available, and if the person is very skillful, he know which point to connect and what to connect, how to connect, and where to detach from the research, so that that model that data can be utilized and can be converted into an final outcome. That was a personalized skill system. Now, what AI is giving is controlled by the digital computer, because throughout a large, couple of decades, we have collected so many data, either in small molecule, large molecule, biopharma, or in terms of clinical research. Now it's available in a consolidated form. Now, once it's a consolidated form, now it's now rewiring our thought process, rewiring our human intelligence, so that how we quickly prompt and utilize those data. So here, when I say how I define AI is a digital computer or a robot which control a large amount of data using a human intelligence reasoning system to learn the outcome very quickly to have an turnaround in the market. Now, this has emerged in 90s, 40s, and 90s, 50s, but the researchers has collected so many data. Now it's available in a large model. Now, what is the challenges that we have in a traditional way? Now, in a traditional way, as I said, is a skill-based system. When I say skill-based system, one person and another person is completely different in terms of technical knowledge. And when it comes to a technical knowledge, the perception changes, vision changes, the key problem statement changes. Whether the yesterday's data correlatable to today data is depends on the person ability to get those things, and get those kind of data in a older period of time. Every time you have to go to the library, you pick off all the books, and then going to the specific page, specific problem, specific words what you're looking for, then you will come to know that you really reach out to the outcome of what you're looking for. And that takes a lot of resources, time, as well as your personal interest to continue for a longer period of time. So here, cost as well as the time consuming process was the key challenges. Now, what we have come up with, like, AI is turning around everything so quickly. Now, when it's turning around so quickly, either a pilot model or a pivotal model, it's giving in fraction of second. Now, if you want anything, you just search it either in the form of ChatGPT or Gemini, or use any large LLM model. At least it gives you a summary. May not be the exact outcome that you're looking for, but it gives you a summary that what you wanted to know and what you are looking for. So that summary is help us to quickly decide that, what really that we wanted to do. Now, with those advantages, what is the thing that was happening in a traditional way? The traditional way is empirical and reactive. Empirical means when you want to put key in the data, if any human error happens, then you have to run the whole thing again. When you want to run the whole thing again, whether you really know that the process has gone wrong, or your data collection has gone wrong, or the key input has gone wrong, so you never know. So in an industry term, we call it as an trial and error experimentation. Without a root cause, you keep on doing it until the right thing that comes up. And that costs a lot to the industry. When it costs a lot to the industry, specifically for the generic industry, money matters a lot, because on a quick turnaround, on a minimum investment, that makes a key to the early market and for the business. Now, what enhances our traditional approach is a data-driven modeling right now. Not only in the Novogen, not only in any of the company, now you have a model where you can key in all the data, whatever you're generating in your from your initial understanding of the product to the level of what pilot model is about. Now everything is data-driven. Now there is no way that you're gonna miss any data. Just you have to key in and put it into a system so that all the wirings of the data will be available. Now, what will happen because of it? Now, in any chance or the medium that you are not remembering those stuff, and you once you wrote it in your experiment book and you skip to write it, and but it's available in the model, then it reduces your experimentation, and it immediately pops up and gives that, Arun, stop it. Now you have already this data. Now you are running around all those variables what you have already studied. Now is the time to concise and precisely look after what you're going to do. So physical experimentation as well as the resource utilization, it's turn around everything. Like it stops every stage which is supposed to be industries to be done on a longer period of time. So with this, the key insight of having the AI in the current industry is reduce the timeline, minimizes the visible experimentation by identifying the potential in the formulation, which is normally known for an incompatibility, any biorelevant issue which lead to insolubility and the connecting issue or stability issue in the throughout the product life cycle. Now, what is the key element that is available in the currently as an AI, so that in the future anything linked to this and how best we can come out of in the industry, you never know when it's gonna turn around. The first thing which comes around is the machine learning. Machine learning is like by human interference, by the human knowledge or the experimentation. You have a large amount of set. Now, when I say large amount of set, as the larger the number of set, that much the conceptuality of having the lower failure is higher. So it's a foundation. Like it learns from the pattern. One person or a one data on a same day, how the experiment has been done. On a second day, the same experiment, how is done and what the variable that has contributed. Now, keeping those in mind for a longer period of time and a longer number of batches or the large number of batches, it predicts easily that what can go wrong and where you have to precisely work on to increase the productivity. For an example, your productivity, or you are working on any problem statement. So this machine learning gives production, specifically in the production, to key insight that what is the key thing that you want to work, and it predicts that it can give an outcome of the product. Second is the natural language processing. Now, when I say natural language processing, is that it's interaction between the machine and the human. When I say interaction between, there are a lot of bunch of data available. Now the prediction or the key prompt, what you're putting into a machine, it's changeable day to day, because 10 number of batches data are available. Now you have one kind of an understanding. Now after 10 batches of data, basis to that understanding, you are designing your experiment, and it gives a large number of data where it is in the favor what you have visualized or you have aimed for, or may not be. and the data set changes. When data set changes from 10 experiment to the 100 experiment, your whole concept of knowing those product is changes. So now with those 10 plus of infinity number of batches, when you key in, then you have to change your thought process of knowing the stuff and interact with the machine and give a prompt to collect all those data and get the things done how quickly that you want to conclude. The next is computer vision. Now, this is a very key thing which is evolving in the pharma industry, which is linked to the drug discovery as well in the compared to the previous session. Everything cannot be measured only in terms of number in pharma industry, because in pharma industry when we talk about drug compound, it has a structure. When it has a structure, it has its own bonds or anything you name it, it has its own function. Now, when you want to predict that thing, how it's going to impact in a pharmaceutical or in your experimentation, then knowing the molecule from the basic, it means structure, you should be aware of it. Now, first thing is interaction of the machine and the human with their language. Now here with an understanding of image and the input. Now I'll put one structure. In that one structure, for example, molecule A, what is an interaction of molecule or the excipient A? When I combine together this structure by visually, now what can go wrong by knowing the by already knowing the process or going to know the process, it gives a complete visual input that what can go wrong. And when you know that what can go wrong, it means you will not do that experiment unnecessarily, because you already know that how is what is the outcome of that experiment. So that's an idea of having the computer vision or the CVA model. Now the final model, which is the deep learning, now is a combined together of NLP and the CV. NLP and CV. Now, once you talk about the natural languages, you can only talk about the natural languages. Once you key in the computer vision may not be feasible. Now, once you have a clear visible, then you have to talk about only visualization or the molecular structure only. How is the better process when you key in the structure, key in your language, and then you can talk to the whole system, like how you want to derive to your conclusion or the formulation development that you're looking for. So the deep learning system is an advance using several layers of complex data understanding of natural language programming as well as the computer system. Now, I'll just go through or summarize one by one how the machine learning and what these four languages which we are talking about. The first, machine learning, is the visual screening, which I said it to you, like it's about the whole number of data, filtered compound. You put it into number of data. When you filter it out, then from there, you know that this is what you're looking for. So the first thing, like virtual screening from those data, this is required, this is not required. Automatically, the machine will gives you that, this is not the trend that you're looking for. It filters out and it gives you only one stuff which you're looking for. Then integrating. Now, when I say integrating, it's a better prediction. Now you have filtered out the large chunk to the small one. Now from that small one, now you're integrating your database. Now whether what is available and what you're looking for, whether this integration matches or not. When this integration matches or that's what you're looking for around 80%, then the enhancement comes in. Now from there, you can pick up what is the promising that you're looking for. Now, if I want to put it into this one into a formulation development, now how I'll put it into. I'm working on a molecule A, and I want to use it in the new development, and I don't know in which excipient that may be incompatible. And if I want to know what is incompatible, I have to run through all the similar or may not be the similar structure of an excipient, combine together the incompatibility study or the compatibility study, which generally we use it in the terminology of industry. Then we come to know that this excipient is not feasible or compatible with this API. Then only we'll study that we'll come to know that after a certain period of time, okay, I cannot use this in my formulation, and that takes time. Now, if I know in a virtual screening itself that this structure of excipient may not be feasible, like my Maillard reaction or anything you name it in the book, if you know that this compound is not compatible with this, that first screening will happen. Now, when this first screening is happen, the next screening is about how much quantity that you're going to use it. It's not about only about excipient incompatibility. The quantification matters a lot. Now, one molecule A with an excipient A may be workable with 10% of the concentration of an excipient. But the same thing, if you increase the concentration of an excipient to the ratio of 1:2 to 1:5, the concentration, when it increases, it stimulates the reaction faster. When it reacts faster, then the incompatibility comes more prominent. So that database will help you to predict that, how much concentration or in which temperature or in which RS that may not be feasible. Now, once I know that this is the concentration that may not be working, then from that concentration, I will be doing my, permutation combination to make my very comfortable product. The second thing, natural language processing, as I said, is an text processing unit. Now you understood from these several texts of your research paper, you have done meaningful analysis. Now you're putting all those 10 papers together in your system. Now within that 10 paper, now you're starting identifying or the interpreting and talking to those 10 paper. What is my understanding of this particular sentence is about? So that's how the text processing and the discussion in the natural language processing is happens. Now, once you know that, then they extract the value information, because how much you want to talk to that data or the processes, that much the outcome it will refine. And once that refination comes, as a researcher, I will go and search more to support those information, and I'll key into that system, and I'll come to know that, okay, now I have to change my perspective, and I have to do some modification in my processes. Then you are identifying that, okay, fine, now doing all those stuff, now I know that this drug and these processes may not be compatible, and I have to modify certain things. Then you are keep on discussing, and then it started appearing in your researches. Now, how computer aided things is working now. In an earlier discussion also, speaker was talking about the molecule and the receptor, how it's gonna bind together or interact together, so that the drug molecule and drug and receptor interaction we will come to know. In a similar way, this computer-aided drug program helps us to know, because you know the receptor, how does it looks like, in what kind of a binding that is gonna happen. You know the molecule, what kind of a structure and how this binding gonna happen. Now putting into a visualization, combine together and put it into a system. Now this gives you the drug behavior. Now this forecast gives you drug interaction. You know that this interaction with this drug molecule, how that's going to impact. Combine together, put it into a system, you will come to know. Now, what will happen because of it? It reduces your drug interaction study when you know that by changing this thing, it is not going to in vivo work. So it reduces the cycle of experimentation and trials, rounds, and predictions outcome, molecule to molecule interaction, molecule to human interaction, gene interaction, all those things you will come to know very quickly. Now, use case application. Now, as I said, in a real world case is not going to be like what you are studying molecule to molecule. In a real world case, you will come to know that what are the different kind of things can happen in around of the molecule. The changes in the structure, changes in the in vivo condition. So AI, by visualizing those structure, when you put it into a system, then it clearly gives you the understanding that definitely this is not the way that you are looking for. These are the probable things which ideally can impact in your formulation, or it can impact in your in vivo behavior. So it enhances the drug discovery. And when I say drug discovery, is not the new molecule here. Either it can help in the new molecule, but the terminology what I'm using here, if I'm using a different polymorphic form, if I'm using a different salt form, if I'm using it in a different processes. So how this is going to be different from the reference product? So that prediction I'll come to know, and that application I'll come to know. So it's very easy for me now. That this is not the path that I'm looking for. Then I'll change my path. Deep learning is a very complex system. Is all about the language model, which I was talking about. I will not go into a lot of IT stuff, but the deep learning is nothing but an NLP as well as the key computer-aided drug program. Put it into it, and then you will start getting in a different model. This is keep on evolving in the system, but this is how it's going to be evolving in the pharma industry. This deep learning system not at morely advancedly available, but somewhere it is applicable. Now, how it's helping in the drug discovery? Now, first thing in the drug discovery is identify the drug target. It's linked to the different molecules or the different structures or different polymorphic form or the different salt. Does this difference in the polymorphic form or a salt form has different biological data? That makes a lot of differences, because we cannot have altogether a different thing which may have a different uh, um, in vivo differences in the body. So to know that, first to know that what is the drug target. So AI helps you if you are using a different salt, different polymorphic form, or into a different dosage form also. It gives you a prediction that okay, now whatever you are changing potentially is gonna work like a reference product. So once you know that it's gonna work like a reference product, then you are on the right track for the generic industry. Now you are not changing majorly anything. Enhances virtual screening. When I say enhances virtual screening, you will come to know that what to use, what not to use. Initially itself, like you identify the drug target, now you put it into a screening, you will come to know, okay, I can use this salt, or I can use this polymorphic form, or I can use with this API or excipient. If no, then stop it over there and just go back to the basic and study and come back. Third thing, aids in molecular dispersion, predicts the structure and the property. When I say predict the structure, once goes in vivo, it changes its own behavior. Now, this is linked to the new chemical entity. When the new molecule comes in the market with a different pharmacokinetic or different pharmacodynamic claims, then this helps us to understand that the design what we are looking for is the same or it's different. Now optimize the preclinical development. Now how it's optimized the preclinical development? Now you know that whatever data that you have generated earlier, you have in the system. Now how much this molecule is relevant or irrelevant? If it's relevant, so you need not to repeat those studies again. And that that shrinks the timeline. Then when it shrinks the timeline, quickly you can turn around the new molecule in the market. And streamline the clinical trials. Now, when preclinical trials, you know that what to avoid in the clinical trial, it facilitate the patient requirement, what kind of a subject that you are looking for, what kind of an studies that you wanted to do, and what kind of a trial that you wanted to do in your formulation, and fine tuning of that formulation that you have to do. All those things turn around very quickly. Now, when all this turn around very quickly, it means you can launch it very quickly in the market. Now, what will happen when you do this stuff? Is like a personalized medicine. There was a question like how is drug discovery will help in the personalized medicine? All the genes, person, people, their requirement, their lifestyle, everyone is different from each other. So drug discovery or the artificial intelligence combining together, it helps to streamline the personalized medicines. Once you key in the data, in a machine learning, and that's it. So every patient's differences will come up, and it will say that Arun, that's not the thing that you're looking for. Maybe today 100 mg molecule is available, but my body weight is not suitable to tolerate 100 mg. So it may say that the 100 mg is not suitable for you. You can have on 50 mg as well. So that kind of a personalized medicine and the formulation can be designed. And once you know that every person is different, so you know that every person has to be treated different. Now, once it has to be treated different, like multiple medicine, like for one person for an anti-diabetic, one person for an antihypertensive, one medication for the cardiovascular system. You are having the lot chunk of tablet together. Now, this kind of a complexity can be reduced when you treat the patients, when you know that every patient has his own medication. So formulation design, probably researchers might be aware of the 3D printing technology, where you can have a different medication in the one pill system itself. So that's a personalized medicine, and you put together into a treatment optimization. Now, what will happen when this treatment optimization happens? So your body will adopt to a different pharmacological system that you're looking for. Maybe you are having a cardiovascular drug, and when you combine with an anti-diabetic together, may not be a similar prediction that you're looking for, or the behavior, the feeling of what you are having it. So can I change the any one anti-diabetic drug when I'm having a cardiovascular drug? Yeah, that's possible when you know all this stuff of an human behavior. So you can change to the personalized medication. Now, once that so happen, now if the person has that appearance when and that combinations is clinically proven, then the whole product drug development changes. When I say whole drug product development changes, then leading to the again going back to the personalized medicine again. What will happen because of having all the personalization medicines together? So it will lead to a whole database of the human behavior, where computationally targeted people of having similar kind of a medicine, we can have analyze, and we can keep that medicine for a key processes to take it to the reliable target identification and put it into an human behavior wherever they want to, then integrate how many patients that may need it, and that personalized medicine can be a group of medicine that it can be driven to. So having a designed for AI to utilize for a personalized medicine, then it can be changed to the group of people as well by having the people's data in a drug discovery. Now, by having this data, how it is helping me in a formulation. Now, in a formulation, as I said, every process is different, every excipient is different, the selection of molecule itself is different. Now, the first thing is efficient selection of excipient. I have discussed about it, what kind of a ratio, what kind of an excipient that I have used. If I know initially itself, then the most of the incompatibility and stability study, I can avoid it later. Like you come to know after 6 months, instead of waiting for the 6 months, if I know in the screening itself that it's going to work in a better way, then I may not be waiting for the 6 months. Improved stability and delivery, yeah, AI helps in demonstrating like how the targeted delivery and the solubility of a system that is helping to absorb in the body, and analyzing and optimizing the excipient ratio. So overall, it's related to the formulation of whether I'm selecting the right excipient for the right process, whether I'm using the right concentration of excipient for that process, or anything can go wrong if I does that. So instead of waiting for a longer period, I may get it in couple of weeks, or maybe instantly also I can get it. So having this concept now, I would like to take it to the demo. I think one of the person has asked in the previous session that is there any tool available in the market which we can use it, and we can know that how these stuffs work. So this is a tool that I was talking how I want to take it to the. Just confirm me whether it is visible.
It's visible, sir.
Okay. So now this is
Farm DE.
Farm DE, correct. So Farm DE is a website or an AI tool. You can say it gives an integration of drug excipient compatibility evaluation so quickly. Now, what it does with rule-based matching. Now, this rule-based matching is nothing but whatever data I have put it into one rule. Like the structure A of anything which is available, and I have a structure of 10 excipients. If these excipients are combined with this structure, I have created a rule that the nitrogen element which is, or the nitrogen molecule which is present over there in the structure, another excipient, if I combine together, then it may that may be an incompatible. So it's a rule-based matching. I have created a workflow and that workflow gives you an outcome that this may go wrong. So this is how this whole incompatibility system works out. So input, you know the compound, means molecule, any API you can say, drug, database match, because the structural analysis, everything is available. Then drug formulation. Now incompatibility comes here. Compound structure, drug compound. When you have a drug database match, it gives you the drug excipient incompatibility data. Now, when you know the structural identification, if you're gonna do it for the new molecule, then based on the rule-based matching, you will come to know what is the risk that you're carrying in your formulation. Now, just for an example, now let's go to anyone. So this database, this is an open database. You can use it everywhere. I think 583 excipients are available and 300 molecule data are available, and
Okay.
it's getting evolved day by day.
Okay. Okay.
So now, for example, now I want to do a drug incompatibility study. I'll go to this one, and if you know, then you can directly put in smiles of a drug, or you can draw a molecule. Now, if you want to draw a molecule, then it's a complex process. You go here, then you draw it, then it will it's a new molecule you draw, and then you put it into a system. And if you are working on a known molecule, then just go to the find drug, and then you search anything. Yeah, like if I want to do sitagliptin, for example.
Sita. Okay.
Sitagliptin. Now, if you go to the sitagliptin, it now is diverting to the drug bank. See, the AI is checking me that whether I am a human or no. Very funny. Now I have into the sitagliptin in a drug bank. Now, how to find a smile? You go here in a download below the structure. You go here and put it into a smile. Now, once you select a smile, this is the smile of the molecule. Now, copy this, go back and put it here as a paste option. Go. Now, this will give you this molecule, which is sitagliptin, has primary or a secondary amine, can undergo the Maillard reaction. And what kind of an excipient to be avoided? You should avoid this kind of an excipient: lactose, glucose, dextrose, dextrase, and acacia. It can form an acylation reaction also. So what should be avoided? You should avoid this kind of an excipient. Again, it can undergo the oxidation reaction. Avoid this kind of excipients. It can undergo oxidation again. Avoid this kind of an excipient. So many. You keep on scrolling, it will give you this molecule can undergo what kind of a reaction with what kind of an excipient, and that's it. Now probably you avoid using it. So this is how the drug incompatibility will appear in front of you. Now, what is the formulation risk that you are carrying? Now, for an example here. Now, again, if you do not know, then you have to follow the same procedure what I have shown you, or else you paste same smile structure of the sitagliptin. Now I'm going to use lactose. Now, for example, in my formulation, I'm going to use a lactose. Now, there you go. Now, let me see what I'm carrying in my development. Is it workable or no? I'm carrying a medium risk. It's showing that I'm carrying a medium risk. Is there any identified reaction? No. Is there any rule-based evidence? Is there? Yeah, the rule-based evidence is the nitrogen atom, nitrite, and the nitrate can interact. Reaction can happen, and that's why it's a medium reaction. So this is how the rule-based system works. Hope this will help us. Now I'll go back to my presentation. This is the one tool which normally in a free database available, any researcher
Okay.
student can use it and utilize for your thesis or somewhere in your development. So it will be helpful. So now going back, what how does it helps in the formulation development? Now one key input variable, I can select a variable input, I can predict how it can go wrong, and I can optimize my formulation. And there you go. What is my outcome gonna be? So by having this all together in one structure, my formulation will be very fast. Now by doing that, so the overall scenario, it reduces the development by 75%. Now AI, you have to use it very smartly. Now this is a normal thing that in the industry you should be use very smartly, specific, measurable, attainable, relevant, and time bound. AI is so vast. You ask anything, it will answer you. It will never ever say that you are wrong. It will never ever say that it don't know. The AI doesn't know. So it's a vast information, and you have to be integrate very realistically and quantifiably, very smartly, so that your integration can happen quickly, and you can get an outcome. Now, what is the disadvantage? Because we are using an AI currently in a very large way, so there are regulatory considerations which is evolving day by day. So once you submit anything to the regulatory agency, so they come back with so many questions because keep on evolving, then there will be a lot of clouds. So integrating with an AI as well as the traditional method is a right now which is going on in the industry, but in the future, definitely the AI thing will take over once the evolution completes. But the only drawback at this moment, because lack of data is not consolidated majorly, and we are regulatory is skeptical how we are using it, because we don't know how 100% we are using it. So there are more and more questions, and that may delay the process because of that evaluation. And as a student or as a researcher, what we should be ready enough, our fundamentals should be very strong, because asking the right question matters a lot, rather being just keep on asking a question and you land up somewhere in a frustration. So that should not be the way. Your fundamental has to be strong, where all the professors and the well-script lectures which are available, they will make you to understand what is the question that you have to be asked, and very precisely. Now, once you know, you are confident enough that in which stream on what you want to do 100%, then learn the basic. AI synthesis is not a big thing. You can keep on practicing and you can get it. Learn the practical things, learn the basic, then put it into a practical, and keep on using the software, modify yourself to ask those questions, and development program which the rule based which I have shown you is a certain rule based system. So you can create your own coding and the data analysis system according to your need, and there you go. You can be on right on bullseye and practice applying AI where there is a problem, and there is a problem, a lot of problem, and there is a lot of scope to utilize how you're going to be. So the basic thing, these are the 5 elements that if you are ready enough in a college level or in a pre starting level, and I don't think so AI is gonna tread you anywhere. And that's it, and I'm closing my session here. So if anything, then you can.
Over to Dr. Kale, sir.
Thanks and congratulations, Mr. Arun Pandey, sir, for this wonderful delivery and contributing. Thanks and congratulations, Mr. Arun Pandey, sir, for this wonderful delivery and contributing to this international conference. So the service on AI in drug discovery formulation development was highly impactful and intellectually stimulating. Mr. Arun Pandyan effectively explained how AI is smoothing and helping in the formulation development research. We know formulation development has always been a complex, time intensive, and knowledge driven process involving careful selection of excipients, optimization of process parameters, and assurance of quality, safety, and efficacy. So the use of AI in predicting the drug excipients compatibility, dissolution behavior, stability, bioavailability, and shelf life was discussed in details. So the session also highlighted how AI supports scale-up and manufacturing process by enabling process optimization, risk assessment, and real-time quality management or monitoring. This discussion helped participants understand the practical relevance of AI in ensuring the product quality, safety, and regulating the compliance throughout the formulation and development life cycle. Overall, the session successfully breached the gap between theoretical concepts and industrial application, emphasizing the transformative role of AI in drug discovery as well as formulation and development. So the session significantly enhanced the knowledge and perspective of participants and was highly beneficial for understanding the future needs in pharmaceutical research and development. So I encourage all the participants to actively engage and ask questions and share perspective so that we can collectively enrich our understanding of the rapidly evolving field. So all the participants, I request to raise some question either by raising your hands or put in the chat box.
Yes, any more questions? I can see in chat. Arun sir, am I
Yeah.
audible?
Yes, yes.
How AI is useful in predicting dissolution of dosage form, and should it should it be considered as a reliable solution with actual trials?
Yeah. Now there are a lot of tools available in AI which gives you the predictive dissolution as well. Now, one or two experiment you do it and put it into the system, and you just tell that I want to change this thing according to the process, because there are a lot of cards available where a lot of process variable are there where it can change the dissolution behavior, but keeping all the factor constant. And only by changing only one element of formulation, either in terms of process or either in terms of formulation formula, it works. And we are keep on working on it as a predictive modeling system, where it gives us that what changes or the experiment that I'm gonna do by changing the formula or the process, how the dissolution is going to appear. Uh, it works wonderfully, and there are tools available. And yes, to answer in short, it predicts wonderfully and basis to the whatever key input available at that moment, by keeping one factor constant.
Thank you, Arun sir. Just one small question is there. What dosage forms benefit the most from AI? I don't know what exactly
Yeah,
it is the question.
the question is very valid, because for class 1 or class 3 molecule, it's very straightforward. If it's going to be an immediate release tablet, then it's
Mm-hmm.
very straightforward.
Mm-hmm.
Even itself basis to your, as the experience increases, then you can predict. if
Right.
if the dosage form is going to be any modified release, specifically a delayed release, or a technology driven, like a polymer carrying spray drying, hot melt extrusion, where a lot of polymer and the process involved, it gives a wonderful outcome. So it's more useful, utilizable when your formulation is more complication. So that's where our time, lot of time goes for a development. So you can quickly turn around with the minimum number of trials. So in class 4, class 2, in an immediate release, and in a control release specifically to the delayed release, as well as for a long term Tmax where Cmax has a complexity, it works wonderfully.
Sir, one more question is there. Name of the tool, please, which will help for optimizing studies.
Optimizing in terms of formulation or in terms of process?
Formulation.
Formulation. So the last tool, the Pharma D, what I have shown you, that that works wonderfully. It gives all the rule-based system where it by default gives you before using it that whether you want to use that excipient and that API together or no, and it reduces almost like 60% of the work, and during the screening of your formulation itself, you will come to know that whether you want to use it or no, and your work is almost done. So use that tool. Is a wonderful tool over there to in terms of formulation. And if it's going to be in terms of process, design of an experiment is an wonderful tool available, either using a software like Design Expert or Minitab. Just put off all the processes, then it will give you that where you are going wrong, and will streamline your experimentation process.
So thank you, sir. One more question is there. How AI is useful in predicting dissolution of dosage form, and what should its reality with actual trial?
I lost the question. There was an echo.
Sir, I can repeat the question, Sam.
Yes.
How AI is useful in predicting dissolution of dosage form, and what should its reliability with actual trial?
It's there. 80% or 85%, I can say it is reliable. It gives you a wonderful prediction. Plus minuses of 5 to 7% you can expect, but it gives you the prediction very well. And right now, uh, there are a lot of regulatory agency which accepts the predictive dissolution as well as structural based prediction modeling data, where you need not to do an experiment, only basis to whatever data available, just put it into a predict modeling, and if that data is very reliable with an validated system, then even you need not to do all this experiment. By doing all this stuff, the regulatory body accepts that.
Arun sir, my question is, how can we use it for screening of new excipient? During
Yeah,
research, many new excipients come up. Say,
correct.
for example, herbal excipients are coming up. How can we screen them up?
It's altogether the processing of or the co-processing of an excipient when the new polymer that you want to build up, new polymer or new excipient that you want to build up, it gives you in the processing science, means the process of what kind of a process that you want to, and whether that co-processing of an excipient will remain stable with together, or is going to be newly synthesized or no. So it's synthesis based design that prediction it gives. So the you can apply the system of drug discovery uh, within process science, means the synthesis science. Then you will come to know whether this combining together will remain intact for a longer period in terms of stability, in terms of molecular structure, and really usable or workable in the pharmaceutical or the nutraceutical industries for a longer time. It should not be like we have designed a new excipient, new co-process excipient, and ideally it is not compatible with most of the excipient. It should not be regularized with one or two dosage form. It has to be regularized in a volume base. So
Okay.
it's synthesis based driven, and it gives you the predictive of whether that structure or that molecule can remain for a longer time in a stability, and the compatibility with another excipient. So it predicts very well.
No, if we don't know the structure of the excipient
Okay.
polymer, say for example, we are extracting some seed gum
Okay.
and trying it as a pharmaceutical excipient.
Okay.
So, can this farm D that type of tools help there?
Definitely.
If
Definitely.
it's a raw gum, without establishing its structure and not going into its chemistry side.
Will be some kind of an experiment needed. It's not going to be like just key in and you will get it. If structures are not available, maybe AI will not, help you in any way, because it
Okay, okay,
needs some key input or the human intervention at this moment to predict that what what we are expecting from them. So to what we are expecting from them, then we have to key in. So without key in, I don't think so that it will able to predict it. At least relatable structure we can give. Relatable.
okay, okay. So as the most of them they are glucomanons or galactomanons. So can that structure help it in?
Yeah, correct. So relevant structure or the nearby
Okay,
structure that will
okay.
predict you that you know what you're looking for.
Fine. So one more question. Can you provide any list of AI tools that we should try in pharmaceuticals
Uh,
for research purpose, especially?
It's in language models. The chat GPTs and all those are interactive language models that will give you the quick summary. But if you
Okay,
want to use specifically for the pharma,
research
then
purpose.
research purpose, yeah, it's all paid one, which
Okay,
the by different people it has been named differently. Same
okay,
PharmaD is available as in PharmaDem with some other company,
okay,
DEM. And
okay,
to predict the dissolution, there is called DGM model, where you can digitalize what you are looking for. And there is one model called visual, where it visualizes the structure of the molecule without drawing that. Once you put it into, it gives you a 360-degree of the
okay.
molecule. And there are more actually. If you ask me, just if you give me an,
You can communicate them by email also. We will put it in
sure.
the group.
There are Maltab, there are SiteFinder, there is WinGPC. What I'll do, I'll put it into a chat box.
Right. Please. So my question, one more question, that how AI can help in regulatory submissions? Is there any chance? Because it's a bulk that we have to submit. So can it help out?
You have rightly mentioned. I think you are top on industry need. I think these questions cannot be randomly asked. A regulatory agency like your USFDA and NPRA, they are really key in where they are putting into an AI where the primary screening is going to happen. So it's not going to be human screening. So regulatory agencies are adopting where all the dossiers of modules, what we are
Mhm.
submitting, is going to be AI-driven screening and immediate responses. So that models are working, and maybe probably by July, the first set of pilot outcome we will come to know. But there are a lot of notification from USFDA and the other agencies where they started really working towards that direction. And
That's really a
there
good
is
part of the story.
exactly that will really turn around the approval time of for the all the generic approval system.
Right. So it will be better for you people as well.
Yes, exactly.
Yeah. Any more questions, please? You can put it in the chat box or directly you can raise your hands and ask the questions. The session is still open for discussions. Any more questions? I think there are no questions in the chat box. The question that was there, you have answered all of them, Arun sir. Now it's a time to deliver a vote of thanks to you. So especially, I would like to thank Mr. Arun sir for sparing his valuable time and being with us. Your hands-on experience in the this thing formulation development has a key role in making this session alive. So you demonstrated with your softwares also, or you demonstrated with your AI tool how we can use them really. And that demonstration, it was a hands-on experience for everyone. So I think your session was quite enlightening and putting a real-time needs, telling the real-time needs for all these pharmacists, pharmacy students, researchers, as well as for the industry people. There are a few industry personnel also. They have joined this session, and they were very keen about your session, especially.
Oh,
So
thank you.
yeah,
Great to know
yeah,
that.
a few senior managers are there. So that was really, the topic was very nice, and you have explained it in a great manner. I would also like to extend my very sincere thanks to the session chair, Dr. R.H. Kale sir, who is the principal of PRMSS Anuradha College of Pharmacy. So he summed up the session with an equal intensity. And Arun sir, really, I also extend my sincere thanks to the management of Novigen, who permitted you to give this session, as well as my special thanks to Dr. Irfan sir for providing us such a great speaker. And I think this is the beginning for a tie-up between Anuradha and Novugen. We will go a long way. Just you can contribute this way by putting some inputs for the researchers. We have post graduations in 5, 6 subjects over here. So we have regulatory sciences, the post graduation, then pharmaceutics, industrial pharmacy, quality assurance. So your inputs will be really very valuable for the students during their postgraduate research as well as doctoral research. I'm very sure. So please be in touch with Anuradha Group. So this is the first time debut entry in Anuradha Group. So please be a member of Anuradha Group. That will be a sincere appeal from my side, and a small request that if possible, please share your presentation after you conclude. Yes, sir. So, Pawan sir, the certificate. Okay, okay. The certificate
Yeah.
for is also
First of
released
all,
over here.
thank you, Professor Unmesh and Dr. Pavan, that they were keep on engaging me to have this session, and definitely my heartful gratitude to Dr. Irfan as well, that he has connected me to this wonderful fraternity of Anuradha College. I was keen to know that most of my seniors who I have in the industry I'm working with, they most of them are connected to this college, and I really appreciate that the effort that you people have made, the changes is appearing in the industry. So the key talents which widely available in the industry is came up from this institution, and they have learned it from you. And I got an honor that I can interact with you as well as my future colleagues over there. And I'm really obliged and thankful to the wonderful management and to all the professors and the people over here. Thank
Uh,
you so much.
One question out of box.
Yeah, please.
Are there any opportunities in Malaysia for graduates or postgraduates?
Yeah,
Indian
definitely.
postgraduates, rather.
Yeah, yeah. In case if they want to reach out, that's what I have put it into my LinkedIn, uh, that QR code and my link profile, so that not directly, at least they can be connected. And if there is anything which comes around during the journey, definitely we will be connected. What I'll do, I'll just put my again the profile, so that people, in case if they are really interested, they can connect with me, and in the future, if wherever the collaboration can happen, that will that will enhance both of us.
Thank you. Thank you, Arun sir. Thank you very much once again. And it was a wonderful session. Looking forward for some more special sessions from your side, your aid, in
Thank
future
you
as
so
well.
much. Thank you
Thank you.
so much.
Thank you,
Thank
Arun.
you so much.
Thank you.
Have a wonderful day.
Dear delegates, now session 2 is over and it's a high time for a lunch break. So sharp 2 p.m. we will meet again for last session by Dr. Anwar Sheikh, AI tools for education and research in pharmaceutical sciences. So sharp at 2 will be here. Please take time to join 10 minutes before the session. So I hope the delegates are enjoying the session. If any comments from delegates, they will be quite welcome. At the valedictory, in the valedictory ceremony, if any delegate wants to express their views, they are open, so you can prepare your views if anyone wants to express themselves. So during the valedictory session after session 3, we will be allowing the delegates to express on one or two delegates. So please, with your permission, please, with the permission of chair, I declare that we will take a lunch break and sharp we will be joining at 2 p.m. Thank you everyone.